Hi guys, this is the cellular respiration video that's about the Krebs cycle. So to quickly review glycolysis, okay, this is the slide from the last one. Just to remind you, glycolysis happens in the cytosol of the cell. When glucose enters the cell, glucose is broken down into two molecules of pyruvate. So glucose goes from being a 6-carbon molecule into pyruvate, which is a 3-carbon molecule. And it costs us a couple ATP in the beginning, but in total we're going to get 4 ATP and 2 NADHs. So we end up with a net gain of 2 ATP and 2 NADHs. Okay, so that's glycolysis happens first out in the cytosol. So next, we're going to have the oxidation of pyruvate and the Krebs cycle. Okay, glycolysis is only the start. We're going to go from glucose into 2 molecules of pyruvate. And it's important to keep remembering that we're talking about 2 molecules of pyruvate, because when we show you how much energy we get out of the Krebs cycle, you're going to have to keep in mind that all those things happen twice because we have 2 molecules of pyruvate. So pyruvate still has a lot of energy left in it. Okay, it's still got 3 carbon in it with all those bonds. So there's a lot of energy left in those bonds. Okay, and we have 3 more carbons to strip off, meaning to oxidize. And as long as oxygen is available, pyruvate will enter the mitochondria. Okay, enzymes of the Krebs cycle do the complete oxidation of sugar into carbon dioxide. All right, and just like glycolysis, the Krebs cycle has a ton of enzymes that are responsible for this. So we're going from pyruvate to carbon dioxide, and we're gaining a bunch of energy along the way. 3 carbons there and 1 carbon there. So we're taking pyruvate, the 3-carbon molecule, and releasing it as CO2, which is a 1-carbon molecule. All right, so here's the same picture that we showed a bunch of times now. So here's glycolysis, and now we're going to be moving on to talk about the Krebs cycle. In the next video, we'll go over the electron transport chain, where we're really getting tons of ATP. So first, let's talk about the structure of the mitochondria and review that. Okay, so the mitochondria, you are responsible for the anatomy of the mitochondria. It's got an outer and an inner membrane, and then these inner folds are called cristae, and then the space in the inner area is called the matrix. All right, that's what it looks like under electron microscopy. All right, so it's got a smooth outer membrane, and it's a phospholipid bilayer, and then it's got a folded inner membrane with these cristae, these folds in it. And the reason it's got all these membranes is because reactions are taking place in the membrane. So by shoving a bunch of membrane into a small space, we can have a lot of chemical reactions happening in a small space. So the intermembrane space between the membranes is a fluid-filled space, right? And the matrix in that is the inner fluid-filled space in here, okay? Right? And there's also DNA and ribosomes in the mitochondria, which is part of what led to the idea of the endosymbiotic theory. So the mitochondria has its own DNA for making the proteins that it needs inside those membranes and whatever else the mitochondria needs to survive. So here's the mitochondrial DNA. It's kind of like a circular DNA, like in early prokaryotes. And the enzymes are floating free in the matrix, and some are membrane bound. So what's critical here, you might want to pause the video and draw yourself a quick picture if you don't have a good one, but you are responsible for being able to recognize the different parts of the mitochondria. And you're also responsible to know that mitochondria has its own DNA. Okay, and what cells would have a lot of mitochondria? I think I've said this a bunch of times to you. Okay, so of course the kind that need to do a lot of energetic work, like muscles. Okay, so mitochondria function. So one of the things the mitochondria do is divide, okay? And who else divides like that? Well, we're trying to get you to make the leap that mitochondria were probably originally prokaryotes that were enveloped. So almost all eukaryotic cells have a mitochondria. Not all. Remember the video that we watched that Kerrigan's group did, that the red blood cells, mature red blood cells, do not have any organelles. But most other eukaryotic cells have mitochondria. There may be one very large mitochondrion or hundreds to thousands of individual mitochondria. How many mitochondria there are is correlated with aerobic metabolic activity. So the more activity, the more energy you need, the more mitochondria. And so, but this picture is really to show you that mitochondria divide the same way that bacteria do. Sorry, I'm losing my voice today. So what does this tell us about the evolution of eukaryotes? Well, it's a reminder of the theory of endosymbiosis, that at one time different prokaryotes enveloped each other and then evolved, so that mitochondria is a remnant, a reminder of an early prokaryote. So inside the membrane of a mitochondrion is a lot of transport proteins and enzymes that are necessary for getting a lot of energy. And what's the advantage of a highly folded inner membrane? Well, I told you this, but this is an important concept here. Definitely one you need to remember. Why do we have a ton of highly folded inner membrane? Well, it gives us more surface area for membrane-bound enzymes and permeases. All right, and the penguin reminds us that form fits function. And so the mitochondria are perfectly designed for what they have to do. So they've got a lot of membranes stuffed in a small space to do a ton of high energy reactions. All right, so now we're moving on to the next step. So right after glycolysis, this is the very next step, the oxidation of pyruvate. Okay, so pyruvate enters the mitochondrial matrix. All right, so giving you a picture of the matrix back here. All right, and the oxidation of pyruvate is this reaction, pyruvate into acetyl CoA and carbon dioxide. And this is the three-step process. Okay, and remember that we have two molecules of pyruvate, so it's going to happen twice. Why do we have two molecules of pyruvate? Well, we started with one molecule of glucose, 6 carbons, and through glycolysis, glucose was broken down into 2 pyruvate, 3-carbon molecules. So we have 2 pyruvate that are going to enter for each glucose. Okay, so all right, 2 molecules of carbon dioxide are released. Okay, all right, so we have 3 carbons in pyruvate. There are 2 carbons in acetyl CoA, okay, and 1 carbon in CO2. So they're saying 2 CO2 because we have 2 molecules of pyruvate, okay? But just to show you how it's balanced, we have 3 carbons over here in pyruvate, 2 carbons in acetyl CoA, and 1 carbon in CO2. And you're probably wondering, well, what the heck is acetyl CoA? Well, it's a step in the oxidation of pyruvate. So it's just a 2 carbon molecule, and that's kind of all you need to know about that molecule. Okay, and along this process, we're going to reduce this. We're going to have this reduction reaction here with NAD going to NADH. Oops, sorry, barking dog. Hang on. Sorry, those are our dogs barking in the neighborhood. Gets my dog barking. You know how it goes. All right, so where was I? Oh yeah, the reaction of NAD to NADH, which is important because the NADH is a nice little carrier for the high energy electrons, because remember when that hydrogen is bringing the electron here, attaching there, so that the whole point of this molecule is to carry around electrons. Okay, and we're producing 2 acetyl CoA, okay, 1 for each pyruvate, and then acetyl CoA is going to enter the Krebs cycle. All right, so sometimes you'll see this oxidation of pyruvate kind of explained as a part of the Krebs cycle, but it's important to remember that it's actually an intermediary step between glycolysis and the Krebs cycle. So this comes between glycolysis and the Krebs cycle, and it's all about converting pyruvate to acetyl CoA, giving off carbon dioxide, and reducing NAD to NADH. So for 1 molecule of glucose, we are getting from this step 2 molecules of carbon dioxide, 2 NADH, and 2 acetyl CoA. Okay, that's why there's 2 here, because we're accounting for 2 pyruvates. For each pyruvate, 1 pyruvate makes 1 acetyl CoA and 1 carbon dioxide. Hopefully that's clear to you. Once we get down to carbon dioxide, that's fully oxidized carbon. We're not going to get any more energy out of that carbon. That's it. Okay. All right, moving on. Okay. Where does the CO go? Where does the CO2 go? Excuse me. Well, CO2, it's non-polar, so it's going to go across membranes, and it's going to go to your veins, and then it's going to go to your lungs, and then you're going to exhale and get rid of all the excess carbon dioxide. All right, so that's where the carbon dioxide is going. Okay, so here's showing you how it's crossing the membrane. So here's our pyruvate, the product of glycolysis. Okay, it's going to be transported with the protein here across the membrane of the mitochondrion. We're going to give off CO2. We're going to convert NAD plus to NADH. We're going to use an enzyme, and then we're going to produce acetyl CoA. Okay, so our yield is this tiny little 2-carbon sugar called acetyl CoA, NADH, and CO2. Okay, so for each pyruvate, that's what we get. Since we have 2 pyruvates, each molecule of glucose gives us twice that. There's the pyruvate. Okay, there's our transport protein. The coenzyme A goes in, or is utilized rather. That's an enzyme that's just sitting in the mitochondrion ready to go, and makes the acetyl CoA. And then acetyl CoA can go on to the Krebs cycle. And what's the point of all this? Well, we're still being driven because we want to get ATP. All right, CO2 leaves, okay, and exits the body through respiration, right? And CO2, we can't get any more energy out of that bond. We can't break down that CO2. It's nonpolar. That's it. And it no longer has any value to the cell, so it's out of there. And it's just going to passively diffuse across those membranes back out of the cell. Okay, we're just accounting for our carbons there. Okay, and there's the oxidation. And there's the reduction. We're reducing NAD plus to NADH. Okay. The Krebs cycle, okay, here's Hans Krebs, who it's named after. Some people get confused and sometimes try to put an apostrophe in there. There's no apostrophe. His last name had an S at the end, so it's Hans Krebs. And right, it's also known as the citric acid cycle, which I'll tell you why in a couple minutes. All right, where does it occur? It occurs in the matrix. Okay. It has 8 steps. Each step is catalyzed by a specific enzyme. You do not have to memorize the enzymes. Okay, and it's a stepwise catabolism of a 6-carbon citrate molecule. Okay. The Krebs cycle came around later than glycolysis. Remember that we said glycolysis was a really ancient process that early prokaryotes did it. Like all cells do it. It happens in the cytosol, right? But you had to evolve to have a mitochondria in order to do the Krebs cycle. So, right, does that make evolutionary sense? Sure it does. All right. Bacteria came around 3.5 billion years ago, and they did glycolysis. Then, I think I showed you this in one of the other slides. Then we started having free oxygen around and started having photosynthesis occurring. And then the eukaryotes came around 1.5 billion years ago doing aerobic respiration with organelles in the mitochondria because oxygen is a requirement for this. All right, so here's the basics. We're going to talk about keeping track of our carbons here first. So we have our 3-carbon pyruvate, okay, that gets oxidized into acetyl CoA, which has 2 carbons. Okay, so then the 2-carbon acetyl CoA is going to enter the Krebs cycle. It's going to get joined by a 4-carbon molecule there. All right, and this is the oxidation of sugars. So then, believe it or not, we're going to proceed with a 6-carbon molecule through the rest of the Krebs cycle. And that's called citrate or citric acid. And so that's why we call this the citric acid cycle. And I'll show you where this 4-carbon one comes from in a second. All right, so this 6-carbon citrate goes through many transitions with enzymes, and we start ditching some CO2, some more CO2 along the way. Okay, and this happens twice for every molecule of glucose. All right, so that's just to show you how carbon is working in here. All right, now let's talk about the electron carriers. Okay, what are the electron carriers that we know about? Well, we know about NADH and FADH2. All right, so as it goes around, all right, there's one NADH that gets formed. And there's another one. Okay, there's some ATP. There's an FADH. And another NADH. And that's going to happen twice for each glucose molecule. All right. So in one round of the Krebs cycle, we're getting a little bit of ATP, some NADH, and and FADH2. So you're probably wondering, well, where the heck is all the ATP? Well, most of the ATP is going to happen in the electron transport chain, not here. So every time the carbons get oxidized, an NAD plus gets reduced to NADH. Okay, what's up? Okay, we finally oxidize glucose. We're taking glucose, C6H12O6, okay, giving off CO2 and ending up with 4 ATP. Well, what's the point? This is like barely any ATP to get out of this complicated process. All right, so let's talk about the electron carriers. They're also really hydrogen carriers. The Krebs cycle is making large amounts of electron carriers, because then the electron carriers are going to the electron transport chain where a lot of ATP is going to be produced. All right, so we're getting NADH and FADH2. So what's so important about electron carriers? They go to the electron transport chain and give us a ton of energy. Okay, and there's our friend the ATP synthase that converts ADP with a phosphate to be ATP. Okay, so let's review the energy we're getting out of the Krebs cycle. So see, we didn't even talk about any of those intermediary steps or tons of the enzymes or any of that. All right, pyruvate. Okay, so from glucose, remember we get 2 molecules of pyruvate. All right, this 3-carbon is producing 3 molecules of CO2. Okay, and we are going from NAD and FAD to NADH and FADH2 so that we have our little oink oink electron carriers there, and 1 ATP. So the net gain, remember this is all times 2 because we have glucose, and glucose will give us 2 molecules of pyruvate. So for 1 molecule of glucose, all right, going through the Krebs cycle, we're going to get 2 ATP, 8 NADH, and 2 FADH2 because pyruvate's going to go around the Krebs cycle once and then again. Right, so what's the point of all this? The point is to generate these electron carriers, which are going to give us a ton of energy. Right, these are high energy electrons that are being transported to the electron transport chain. Okay, what's the value of the Krebs cycle? So if the yield is only 2 ATP, then how is the Krebs cycle really an adaptation? Like, why was it successful? It seems like a lot of work just to get 2 ATP. Well, even though we talked so much about how awesome ATP is, these electron carriers have a lot of value because you're going to see we get tons of energy from them. Okay, so the reduced molecules really move the electrons, okay, and it's moving hydrogen ions, and those can all go to be used in the electron transport chain. And the reason they're in these piggy banks here is because they're like money in the bank. These are high energy electrons to be used later on. Okay, what's the point? The point is to make ATP. Okay, and how do we do that? We use the ATP synthase. Okay, set up a hydrogen ion gradient, allow the hydrogen ions to flow through, powering the bonding of phosphate to ADP so that we get ADP plus a phosphate to get ATP. So remember that ATP synthase is an enzyme. It's a little machine that converts ADP into ATP by bonding a phosphate to it. Okay, and so those are, there's our ATP. But have we done that yet? That's the next question. All right, no, we're not done. So that will be in the next video on the electron transport chain. I hope this was helpful, and I will see you soon.